Good afternoon, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and it is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multiomics for guiding personalized medicine, where each Friday we will enjoy presentations from a wide range of leading thinkers about the issues, promise, opportunities, and hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic methods along a wide variety of data analytic opportunities as they present for data science, deep learning, AI, and spatial modeling. Today, we are particularly pleased to announce that our 2024-2025 Biomedical Data Science Innovation Lab application cycle is now open, and we hope that you might help us by encouraging junior faculty members working in various areas of omics-related biology as well as clinical medicine to apply. And aligned with this year's theme, we are eager to receive applications from folks working in multi-omic data analytics, computational biology, machine and deep learning, spatial statistics, network analytics, graphical methods, as well as software design and user interfaces. Participants selected for our special Biomedical Data Science Innovation Lab program will utilize the content from these seminar presentations as essential material in culminating in-person manuscript and grant project development, to be held as a workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We are particularly excited for this program where junior faculty, biomedical researchers, and computational experts will develop joint novel projects on the topic of quantitative approaches and applications for spatial multiomics data types and their use towards personalized medicine. For today's seminar, I am delighted to welcome our speaker, Professor Bill Basener, from right here at the University of Virginia. Bill is a professor in the School of Data Science with a joint appointment in the Department of Systems and Information Engineering. He has authored research publications in machine learning, spatial signal processing, image processing, dynamical systems, game theory, ecological economics, evolutionary genetics, and other applied mathematical fields, as well as having written a textbook on applied topology, where he has developed multiple commercial patents and enjoys a very rich research program. The methods and software he has developed for processing of images and the hyperspectral imaging have become the gold standard in the field, used for processing of millions of images by dozens of organizations. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Professor Basener's lecture today is entitled Making Predictions from Simulations of Complex Temporal-Spatial Biological Systems, where he will tell us that biological systems often revolve around complex interactions which can vary over time and vary by location. This can make cause-effect inference difficult, and the challenge is increasingly difficult when the data itself is difficult to obtain. In his talk, Bill will present the use of Bayesian inference, which can provide probability distributions for input parameters, leading to novel and interesting outcomes from spatial and temporal data. And he'll present a variety of examples using models of lung fibrosis and ecological-economic civilization models. Such computational models may serve as inspiration for spatial approaches in large-scale omics data types. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions via the chat feature in their YouTube sessions, and I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of Bill's lecture today. And with that, welcome, Bill. We are certainly very excited to hear your presentation today. Looking forward to it.
Thank you very much for inviting me, Jack. It's a great pleasure to be here. So what I'm going to talk about today is how do we make predictions from simulations of large, complex models of data. And so here's a main problem. This was a work with PI Shane Pierce Coulter, also here at University of Virginia. And the problem was as follows, shown on the screen. We're modeling human IPF, which is idiopathic pulmonary fibrosis. And I'll just apologize up front. I'm a mathematician and data scientist, so I'm not going to try to frame all of the biomedical components here correctly. But so fibrosis is a buildup of collagen and stiffness in the lungs. And the goal is to understand what causes this and really what parameters of the underlying systems that cause the fibrosis are contributing to this. So we have slides of lung tissue, which are shown here. Let's see, right there. And the question is, what types of parameter values or underlying parameters for the underlying system that generates this, the cell interactions, result in this type of buildup. And so on the bottom part here, we have a diagram of what we're going to be using to model this, which is an agent-based model. So what we really do is we have a simulation of a small component of lung tissue, which is what's shown in this box here. This is healthy lung tissue. This is after 12 months of building up fibrosis, and then this is after 24 months. So the pink or dark pink here is the collagen that's built up in our model, and we want to model every individual cell in this square of lung tissue. So we have a large number of cells. They're moving around, they're depositing collagen, they're pulling up collagen, and what we want to understand are what parameters would you put into the model that would give you agreement between the output of the model and the observations on human lung tissue. There's a little more look at what we have here. This is really the differential equations model. And as you can see, this is a large, complex differential equation with lots of interacting parameters, right? The parameters are on the top, the outcomes are on the bottom. So think of reading it downward. And this was developed by Jay Saucerman here at UVA, and is used by the Pierce Coulter lab to model each individual cell. So what we're going to have is each cell gets modeled. They're going to be moving around and depositing and pulling up collagen. And so I'm going to step over to the software we use for modeling this. And really, my goal today is to provide some help putting together, if somebody wants to use the techniques we're doing here, making them accessible. So understandable and accessible. So this is our model. This is NetLogo. And free download software for doing these types of simulations. Excellent software. So what we have here is a window that's going to show the result of our simulation over time. We're doing what's called agent-based modeling. So each cell over here on the right side is an agent. As time passes, they're going to move around. Their interactions, their function for each cell is going to be governed by that large differential equation that we had. And we have two types of cells. We have the Thi1 positive and the Thi1 negative. The Thi1 positive are the orange, Thi1 negative are the blue, and we can modify our initial conditions by changing the sliders here. Actually, I want to keep that at 10. And then if we run the model, we hit go, and then we can observe what's going on. And so there's a number of parameters here. All the green boxes are different parameters. And so the question is, out of these 10 parameters or so, which parameter settings and which values would generate results that would deposit collagen and lead to fibrosis in the same rate as we see in human fibrosis? And so to do this, what this effectively comes out to modifying these 10 parameters over and over, and then letting this run sufficiently long, and repeating that some number of times, say 10 times, and see if those 10 outcomes match what we see in 10 human patients. That, as you can imagine, is impossible to do on the basic, kind of as I described it. So we need some more sophisticated or automated methods for doing that. So Here's that differential equation. Here's the screen that we were just looking at. And two of the parameters that we're going to be focusing on involve the rate at which collagen is depositioned, call-dep, and call-deg, which is the rate at which the collagen is degraded by the function of the cells. Okay, so let's talk a little bit about Bayesian inference. So what Bayesian inference does, it allows you to take some parameters and then put them into a model. Now, typically the model is might be something like, I have 2 bags of marbles. One bag is 50% white marbles and 50% black. And I have a bag of marbles that's only black marbles. And so my parameter might be the selection of bag that I'm drawing from. The model might be, I pick a marble from whichever bag that I'm dealing with, and then that would output either a white marble or a black marble. And so that would be my output. And I would use that in Bayesian, using Bayes' theorem, to estimate the probability distribution for that set of parameters by comparing it to my data. Right. And there's Bayes' theorem. There's some really beautiful formulas here that I love, but I avoided them because we're not going to use them very much because we're doing simulations. We don't have the nice, tidy, perfect world of a bag that's half white, half black marble. So we have a 50-50 probability, and a bag that's all black marble. So we have a 101 probability for getting a black marble that we would like for the questions that statistics professors like to torture their students with. We have a situation where the model is a complex biological system, or it could be a complex physics system. I do a lot of these with things involving physics, but it's a complex system where it's not deterministic, right? There's randomness, there's walking around, there's interaction with different agents. In the physics system, there's different bouncing around of photons, interaction with different molecules, generating spectra. And so what we do is we really care about connecting these parameters to the data as they get processed through the model, right? Input parameters into the model, we get data. Typically, we're dealing with things where the model is very hard to compute, right? So running a full simulation through what we were just looking at with NetLogo, or running maybe 100 simulations and comparing them to 100 human patients, and then comparing the output. So what we're going to be using is something called approximate Bayesian computation, ABC. And what this does, we can have a prior, which we haven't talked about, but that's typically, you could think about that as before we start working, we have some sense of what those parameters should be, right? So the rate at which collagen gets put down and the rate at which collagen gets pulled up, or degraded by the cells, you have some bounds on that, right? They're between some values. That's a decent prior, right? So you can think of it, if you're used to dealing with priors, as a uniform distribution on some range of values. Then we have our experimental data, and this case, this is our observation of rates of fibrosis and how quickly the collagen accumulates in human populations. And then we run different sets of parameters, or randomly select different sets of parameters, run them through the simulation. And if they match the experimental data close enough, then we kind of add them to our understanding of what the probability distribution is for the parameters, right? So what parameters could lead to things that would match our observed data. So I'm going to step over to some software. And what we're going to do is we're going to run here we go. We're going to look at some data that's been run through Kaggle. Now, Kaggle is an extremely useful online community of people doing data science, machine learning type methods. And so you'll find here, they have an environment where you can upload data and you can run code, whether it's Python or R or whatever, and then distribute the results in a notebook, and other people can come and learn from what you've put together and share and run it on their own. So if you make an account in Kaggle, you'll be able to go and run and modify this this Python notebook that I'm going to take a look at here together. So this is how we would run the software for doing the simulation. So what I'm going to do is I'm going to start with a simulation that's probably pretty familiar. So this is a Lacta Volcera predator-prey model. And so we'll talk about how to set this up and how to do some of the coding to set this up. And so in the beginning part here, we do some imports to import some of the packages we're going to need. And the one that's might be that's specific to what we're doing and not generally a Python package is PyMC3. And so this is a Python module for doing Monte Carlo MC simulations. And it has a good acceleration and use of graphics cards. So, okay, so here's our model. We have 4 parameters. A is a growth rate of the prey. B is the harvesting rate. And so here's our differential equation. And then C is the death rate for a predator in the absence of prey. And then we have D times B, which has to do with how quickly the predators are going to catch the prey and eat them, right? And so the specifics of the equation don't matter very much, but we set up our differential equation here, and then we make a competition model, which involves this. And so what the competition model does, you put in values for A and B, and it will run this differential equation and then figure out what the future is going to be. So think about this piece right here, this function, as running that simulation, that agent-based simulation. In this case, it's a differential equation. We don't have all the spatial complexity and randomness that comes from the agent-based model. And now we're going to generate some data, right? So we're going to use this to simulate our data. And right here, we have our observed data, right? And so this situation, we're thinking about the prey is shown by the blue squares, blue X's, and the prey increases while the predators are small. Prey reaches a peak. Predators start increasing because there's food for the predators and they increase in population. Prey goes down, and then we go through these cycles. And you can observe here that this data is noisy. And so the type of question that we want to ask here is, how do we know what parameters could have generated this data? Really, we want to answer this in a more sophisticated way than just saying that. We want to say, what's the probability distribution on the parameters that would generate this data, right? So that would allow us to say, we know a probability distribution on the parameters that generate the observed data. That can help us to, it understands the system better, but also we can look at other cases and look at the parameter values there and say, will this eventually result in what our model did, or what our, in the fibrosis situation, will these other parameters be likely to result in the fibrosis? Okay, so here's where the approximate Bayesian modeling comes in. It's in this function here. So PM is PyMC3. These are our priors for the parameters A and B. And I know this is predator-prey model is a fairly simple model, but it's one where we can really see how the approximate Bayesian computation is coming into play. So A is the growth rate of the prey, and we're approximating it with a half normal distribution, so it has to be positive. And then B is the rate at which the prey gets eaten, or has to do with the harvesting rate. And so in this simulation, this builds up a simulation. We feed it the computation model. And we have to give it some error, some rejection. In this case, the rejection is this epsilon. And it says, if the result of our simulation is close enough to our observed, then keep those parameter values. If not, we're going to reject those. And that's also where this kernel equals ABC comes into play. Okay, so when we run the model, this is what we get. We get a probability distribution for A, which is the growth rate of our prey, and a probability distribution B, which is the harvesting rate, or corresponds to the harvesting rate. It's one piece of the harvesting rate. So it has to do with how readily the predators will catch the prey. And so we can get a couple of different views of this relationship between A and B, and I apologize. It looks like I labeled them C and H in the plots instead of A and B. So this is the values for A, which would be the growth rate of the prey. And then we have on this axis the harvesting rate. And so you can get the sense of the values that were likely. So this is the density here is a histogram, a 2D histogram, showing the probability distribution for how likely these different values were to generate the observed results. And this is actually Bayes' theorem. It's an original discovery, had to do with doing this type of simulation, looking at planetary motion. I think it was Jupiter or Saturn. I should have had that prepared. I apologize. So it was one of the rediscoveries of Bayes' theorem, or one of the original applications. And so this gives you another view of what that probability distribution is, this relationship between A and B. Another nice plots. And so this plot here shows us the results of the different simulations for the predator-prey model. Those are the lines that were close enough to the observed data, right? The orange dots, that we could say, yeah, this is a possible simulation that could have corresponded to that data. Here's a more complex model, which is in the same sort of category of models. This is a model for the human population on Easter Island. This is a model I did, I don't know, 10 years or so, maybe even a little longer, looking at, this is an anthropology model, trying to model what happened to the population of Easter Island or Rapa Nui, right? So there were a group of people who landed on the island in about a population of 50 people on a couple of boats. They sailed a long way. The island's very isolated. And over time, right? So this is the time axis. This little blue X represents where the people landed over time, maybe 1,400 years or so. Population grew large, built the large statues that the Easter Island is known for, and then crashed and declined. And so this picture here is very sparse data, right? We have 4 axes, and they come from anthropology and people, I would say, guessing, digging in the dirt. I mean, it's more than guessing, but estimating based on what they find digging in the dirt, what they think the human population did, right? So we're going to try to estimate parameters that could have resulted in this sort of system. All right, and again, this is, I think, it's a much simpler model than the fibrosis model, but it's one where we can see the approximate Bayesian computation coming into play. And we can also see that when you use the approximate Bayesian computation, one of the nice things is you don't have to get buried in the probability distributions and the Bayesian inference and the formulas, which, oh, by the way, I love. It's beautiful like a sunset, but it's easier to get into the place of using it from an applied area if you don't have to go through all those pieces. So A is the average growth rate for pre-industrial revolution human population. C is the growth rate for the resources. In this case, it's trees are the resource. So it's a group of people landing on an island. Originally, there were enough trees to support 70,000 people in this model, in this set of parameters. And then H is the lifespan, one of the lifespan of a person. And then H is the harvesting, actually, H is the harvesting rate. It's how many trees an individual person would harvest. And so here's our initial conditions. This is the length of time we're going to run for. Then we set up our differential equation. And then what we're going to do is we set up this function for the modeling, and we're going to model with respect to the parameter C and H. So we come down here. Here's one possible solution, and this is what you would typically find in the anthropology literature prior to this work that we're looking at here, where somebody would say, hey, this is what I think could have happened, and give one simulation that gives you an approximation to it. So this approximate Bayesian information, this box does the ABC modeling, where we set our priors on C and H, And for these, we have a uniform probability distribution that says C is the growth rate of the people or of the resources, and it's between 0 and 1. H is a harvesting rate between 0 and 1. So this is a prior, but it's really just setting our bounds on our rates. And so we do our ABC, approximate Bayesian computation modeling, and we get C, the growth rate for the resources, is between 0.048 and 0.64. This is really important for this situation because it bounds the type of palm trees that could have been growing on the island. There are a lot of anthropology investigations into the seeds and the type of trees that were growing, and the growth rates for those different trees are known. And then this is the range for the harvesting rate. And this corresponds to how, if you are harvesting a resource, you would want to pick your harvesting rate so you're not in the range that would lead to the collapse, right? So this is a economics management type solution looking at the harvesting rate. So that's a harvesting rate you would want to avoid if you were on an island with those trees, right? And so here's, I don't know how many simulations we have here, but here's our simulations, maybe 50 or so, that we're showing that show the increase in the population of people, and then the collapse, and then the steady state of the population of the trees, and then their collapse. And then one of the extensions we did on this model, oh, and this is this is really an important thing that we get to, right? So this is really the goal with this type of work. It's what is the probability distribution, or what are the values for these two parameters together, the C and the H, that would give us, in this case, a collapse of the civilization. So values that are along this diagonal and maybe a little drop to the right, those are the values that are required to get the collapse at the time that we saw in the data, right? And so you get a pretty strict restriction on your pairs of parameter values taken jointly. And so this is called a joint probability distribution because it's joint on these two parameters. And so that's a really good picture of what the goal is, right? Understand which values of these, actually pairs of values together of these two parameters, could lead to a population collapse or whatever we observe in our actual data. Right? Different ways of viewing that. And here's a part 3, which I'll jump to the end. And one of the nice things here is you could build up your models about as complex as you want them to be. In this case, this was a hypothesis from a researcher at University of Hawaii that it's really a rat invasive species that was aboard the boats that caused the population collapse. And so we can add this population of rats in the model. It's not very hard to set up the differential equation. It is hard to do rigorous stability and equilibrium point and long-term trajectory analysis in an analytical perspective on that differential equation. But with the ABC modeling, we set up our priors for our 3 parameters. We set up our ABC model, and we get posterior probability distributions for the values of C and H and F. And F is at the growth rate of the rats, right? And so the rats would gnaw at the trees. F might be the growth rate of the rats, or it might be the rate at which the rats, yeah, it has to do with how the rats impact the tree reproduction rate. So the rats would eat the seeds and eat some of the seedlings and reduce the reproduction rate of the trees. And so we set up our model, and now this joint probability distribution between C and H, we get that. So H allows to drift up into this area with the presence of the rats. So you could take this same framework and build as complex of a model. It could be a differential equation. It could be, as we were looking at with, coming back to here, in this fibrosis work, right? This model here that has 10 different parameters. And so you have this joint probability distribution in 10-dimensional space, which is really complex, really hard to sample and understand the distribution and the interaction between the different parameters. And you could run this over and over again, right? And this is that fairly complex differential equation for each of this 100 different cells. And then we're evolving that, and the cells are laying down and pulling up collagen, and they're moving across this slice of tissue, simulated slice of tissue that we see here, and interacting with each other and with the results of what each other did. But this framework of the ABC modeling allows us to repeat runs of this and then model the parameters that meet the observed data. And so this is my last slide. This was the result of our work, and this involved a School of Data Science master's degree capstone. So I should call out the students for doing great work on there. And so we have these three parameters: K-dep, which has to do with it's a coefficient involving the deposition rate for collagen; K-deg, which is a coefficient rate for the degradation of collagen; and TRT, which I apologize, I don't have a proper way to describe the TRT parameter. But you can see that to meet the human IPF results that were observed in the human population, you want sets of parameters that are in the lighter color areas. And so those are the parameter values that are more likely to result in the fibrosis accumulation that we saw in the human population, or has been observed in the human population in data sets from other researchers. And on the back here, this is the probability distribution with the different shading was drawn by a Python package that fills in based on the results of the simulation. So that's about everything I have to share. So I first want to say thank you very much for the invitation to come along and talk.
Bill, yeah, thank you. Thank you for presenting on this. I mean, this is really interesting because as you were talking, I think, many people are probably familiar with Bayes' theorem and Bayes, kind of calculating your from your marginals and conditional probabilities that you can like compute other probabilities using that approach. But really, you kind of showed that the use of Bayes almost over time also seems to have like an inference engine capability. Would that be a safe thing to kind of comment on?
Yes. Yeah. So Bayesian can be used for inference on giving you a probability distributions on your parameters. So we use it in class, and we sometimes in courses, or when you see it in one place or another, you don't get a full appreciation of the flexibility of Basener. So when I teach my Bayes machine learning course, I talk about machine learning models. They come prepackaged in a lot of ways, and they'll give you a prediction based on inputs. Bayes' theorem, or the whole Bayes machine learning framework, is more like a whole set of Legos where you build your own tool and your own toy from scratch, and you can get a lot more flexibility out of it. So in this case, we're using it for inference on the parameters.
And you talked briefly about kind of Monte Carlo simulation and what are some of the things that one would need to know in order to do a Monte Carlo simulation? In fact, what is meant by Monte Carlo simulation? What are some of the distributions one could potentially sample from that, and what ones do you find helpful, or does it depend on the context?
So with a Monte Carlo simulation, we are taking we have a probability distribution for something, and we're repeatedly taking a draw from that and doing some computation based on that draw. And so here, take a look at another Kaggle notebook, which I think will be both interesting for the Monte Carlo question and also by the way, Kaggle is a great place to tinker around with. So this is an example using a Monte Carlo simulation, looking at analysis of mining disasters, right? So what we have is this data, which is the number of disasters per year in mines over an expanse of years from 1851 to 1962. And in this case, we're going to see something interesting here. This is an analysis of switch point, what's called switch point analysis. So we're looking for a switch point where the rate at which disasters occurred goes from a lower value, from a higher value to lower value. And if you look at this data, right, there's a lot of high values early on, low values later on. And the question is, when did that switch occur? And really, we want a probability distribution for when that switch occurred, right? So there's an underlying system for mining disasters that went from something high to something low. And so we used a Monte Carlo here and Bayes' theorem to estimate the probability distribution for when that switch occurred. And one of the important questions is that there was a change in the laws in a year, and let's see, I don't see the year here. But the question is, did the reduction in mining disasters occur at the same time as that change in law? And so here's a plot of our data. Here is the prior distributions. So the switch point, which is the year that it switched from high to low, we're allowing that to go from 1851 to 1962. And so the Monte Carlo simulation is going to pull a date from that range. Then it's going to say, okay, if that date was a date where a switch occurred, what would the early rate and late rate be? And then it's going to compare that to the data and say, how well does this fit the data? In this case, we can, when we say how well does that fit the data, we can frame that in the computation of a likelihood and then use Bayes' theorem with likelihoods to compute a probability for that date. So because we have data and we have a model with distributions, so we can approximate that. And so the switch point is what we're doing our Monte Carlo simulation. We're randomly drawing dates in that range. Then the early rate, which is the rate at which these occurred before the switch point, and late rates, the rate at which they occurred after the switch point. So the interaction between these different values. And the rate for each year, the rate at which mining disasters occurred is here. And we say the rate at which they occurred, that's the averaged rate. It's the actual number of disasters is drawn from a Poisson distribution, whose mean rate is that average rate, which goes from the before value to the later value. And here's how we set up our Monte Carlo run, where we're going to take lots of values of what the switch point date is. In this case, I think 10,000 values. We run them through the disaster model, and then we get a probability distribution for the year for that switch point. And so in this case, the dates are a little jumbled in, but you can get a sense of when the year was. This actually does this imputation on the missing data. There are a few nans, so this will estimate the most likely values for those missing values. And this gives us a probability distribution for the early rate, which is about 3.1 or so. So it's telling us before the switch occurred, there were about 3.1 disasters per year. But we're not sure what the exact value is because this isn't generated by, the data is not generated by something with a fixed value. There's estimating the uncertainty around that value, which is one of the main things that Bayes' theorem gives us. And then here's the late rate, which is the rate of disasters per year after the switch point. And so we see that the switch point, or the disaster rate, went from about 3.1 down to about 0.9. And this PyMC3 gives us a really nice framework for understanding like confidence intervals around those different values. And it gives us a nice plot of the black dotted line is the maximum probability value for the rate of disasters in each year. And then the orange gives us the bold orange line gives us the most probable date when the switch point occurred, and then the shaded area gives us a uncertainty around that line. And so there were safety laws passed in 1891, and we see in this case that falls in the range for the orange, suggesting that the rate occurred around 1891, and so that would be a reasonable cause.
Bill, one question is in addition to the distributional parameters, can you take into account like a temporal lag in a model like this? Like, for example, in like you said, the 1880s, for example, there was some laws that were changed, but very often there's a lag, right, between the time when the law changes and when the safety, improvement happens, for example. It might have taken 3 to 5 years, maybe. Can you model that as well, or can you have the model kind of give you an estimate of how long that lag might have been?
Certainly. Yeah, anything like that where you want to build, you have a increasingly complex relationship between your parameters. It's one of the nice flexibility parts of Bayes' theorem and Bayesian methods is that you can build that into your relationship between your parameters and estimate that amount of lag. Yep.
One other thing is that you've kind of mentioned how some of these things can be done over time, which is very often of interest in biology. But one of the things which is also kind of interesting is over space. And so you kind of showed with some of your agent-based modeling that you can model things over space. I'm curious about the use of kind of some of these Bayesian methods where things may vary over space depending on tissue density, or I don't know, there's some particular presence of a, I don't know, a chemical or something in a tissue sample that has a gradient to it. At any rate, it's changing the underlying spatial properties. Can you model those as well?
Yes, certainly. When we set up these sort of 10, now I guess it's 9 parameters here in the green box, in the green with the green sliders, you could think of those as parameters into this location, but you could also think of changing those as changing a parameter model, a situation in a different part of the body, a different type of tissue. So as we think about the parameters, they're, things that govern the situation, but they could also be a location or other, any factors that would impact the environment.
I ask this question because I know a little something about you and your other life where you're doing kind of like remote sensing kind of activity, or you're using like LiDAR technology to look at, say, the density of foliation on a plot of land, for example. And I would presume that the models that you build when you're looking at those might need to have some sort of spatially varying prior, if you will, and that you take those into account in your modeling. And this would not be too dissimilar from, say, taking a look at a cancer biopsy and noting that, there are varying parameters across the spatial profile of that cancer biopsy. Would you explain a little bit about how you do that when you're thinking about things in terms of LiDAR or satellite imaging?
Yep. So one of the areas I do in the remote sensing world is the hyperspectral imaging, where you collect an image over an area in the ground. Each pixel has a full spectra for the location. So digital image we're used to has 3 bands. So we see the red, green, blue. But the hyperspectral images have hundreds of bands, and you get a full spectrum. So you can do spectroscopy and identify the chemistry of the materials there. On the Bayesian side, what we do, we use the things that we are looking at here is we have the spectra for the pixel. We know that occurs from the interaction of light in different materials and chemistry and chemical bonds that were present in that material, but we're unsure about the exact mixture of what was at that location. And so this same Bayesian inference we're using here on the parameters for collagen deposition rate, we use on the understanding the pixel, and the parameters are the amounts of different chemicals that would be present in that pixel. And in that case, if you find something with a high ammonia content and you're in the mountains, that high ammonia content is likely to be due to minerals, feldspars, ammoniated feldspars, or buddingtonite, which in the mineral world, those are things that occur that are indicators of underground volcanic type activity. If you find ammoniated things in the water, then they're likely to be concentrations caused by over fertilization and drought conditions that concentrate the impurities in water in shallow pools. So we build priors into the models that say, based on whether you're mountains or water or somewhere else, the things that we see in the minerals have different, or in the pixel and the spectra have different implications for what's going on at that location, or what the kind of bulk type material at that location is. So same methods we're using here. That's set up as like a different parameter that feeds into your model. And that additional parameter is a parameter, just everything we're doing here, but that additional parameter is dependent on spatial location.
And Bill, I'm kind of a fan of and intrigued by agent-based modeling and the example that you started with. It's just really kind of interesting to see these, little uh, objects, floating around or running around, and they're depositing the collagen in this case. And there's other ones that come around and eat it up, and you can manipulate the various parameters. What are some other examples from biology that you might be familiar with?
There are a whole set of built-in models. So there's a lot of genetics models and evolution models. So you can model genetic drift. These models don't have in NetLogo, they can do the agent-based modeling part. NetLogo is really good at that part. These don't have the Bayesian part. We write the code in Python that sits outside and then calls the NetLogo code, allows the NetLogo code to run, gets the results back, and then iterates on that. So there's a terrific variety of models that you can run. All right, so here's a bunch of shepherds taking care of their sheep.
Herding efficiency. I like it.
Yes.
Because I mean, you can imagine that you have something like, T cells are, there's an infection. T cells are now like deployed to kind of go and gobble it up. And if you had something like an HIV, for example, where there was alteration in the ability of certain cells to go in and do their job properly, that you could get a runaway infection, and that you could model those processes, using an agent-based type system like this. That'd be fascinating. And I'm also curious because these are complicated, right? You've got multiple things moving around. You've got these partial differential equations. You've got Bayesian Monte Carlo simulated things. These things can get complicated pretty quickly. I'm curious about any assumptions that are made concerning the like stationarity of these systems over time. Are they ergodic, or are there other stochastic processes that are part of them? How do you contend with those?
That's a good question. You could build in any components you wanted to, right? So you could have a couple of, in this case, I've got a square, but you could have a couple of different pieces and they're doing their things, but there's some feedback loops between them. So infection in one part of the body, how does that interact with things going on in other parts of the body, where you model them separately in addition to their interactions. You can put into the code. So here's the code for modeling those. You can put into these any random variables that you want. And when you repeat your runs using the Bayesian methods, you'll learn how those random variables impact the outcomes.
And many people who are going to be interested in our biomedical data science innovation lab this year will probably be interested in things like gene expression and how genes like turn themselves on, express proteins, and then eventually turn themselves off, or don't. And this is you're going to be something of you've got a location on a genome, for example, of a particular gene, but the genes in one part of the body may be expressing a particular protein, whereas in other parts of the body, they're not. And I'm curious if there's any examples that are like that you're familiar with.
There aren't any that I know of, but that would definitely be something you could build in if they're not already present here.
And this one's the one you're showing us is kind of like a virus model. Is that what we've got here?
Yeah, yeah. And you can build in in this case, the spatial dependence of people randomly bumping into each other and how that influences the spread of the virus.
I imagine this was kind of stuff that, oh, maybe about, oh, say 4 or 5 years ago would have been really interesting.
Yeah, sorry to,
You're late. You're
bad
late. You're
memories.
late, Bill. Bill, how would you make a recommendation for people who are they're intrigued by some of the things that you've talked about? How could they get started? Is NetLogo a great place to get started, or what else might they need to know in order to begin using some of these tools?
Um, NetLogo is a great spot for the agent-based modeling. They have, as you can see, terrific models to go through and take a look at, and then to start modifying. On the um, Bayesian and model forming part, Kaggle's a great resource. Kaggle has a lot of things where if you put something into a search, you can find um, people that have done things that are similar and then commented on it. And there's a lot of feedback on a lot of input on different ways to optimize and do well with the models. There's a fair amount of PyMC3 material on there. Of course, PyMC3 has its documentation website. But I find for the PyMC3, which does the Monte Carlo Bayesian modeling, a lot of the Kaggle notebooks, if you open those up and start tinkering, you can modify those to get what you want. So the PyMC3 Python Monte Carlo methods are, I find it often easier to modify things that are already present to build those.
This is fantastic. I love watching all these examples, Bill. They're great.
Yeah, you can spend a lot of time looking at these. It's really amazing with the NetLogo, the amount of interactions. It's very creative because any interaction that you can write down formulas for that has a spatial dependence or little pieces interacting with each other, the NetLogo can do a pretty good job.
One thing which I was going to ask you about is with some of these agent-based models, they many times when I see them, they sort of remind me of Conway's Game of Life in a matter of speaking, right? Where there's a lot going on. It looks very complex, but they're based upon very simple rules. Can you maybe explain a little bit about that and your experience, and if that is something that, again, it's this kind of notion of parsimony, right? It's simple, a few set of rules, but if you set them up right, you can create really complicated behaviors. Can you comment on that?
Yeah, my as a grad student, a lot of my work was in dynamical systems in chaos, which is often in fractals, and how you start with a simple rule and you repeat that simple rule over, and you wind up with things that are very complex. But it's a infinitely complex because if you zoom in and zoom out with Conway's Game of Life, you see these patterns on different scales. But it's also, in a sense, like you were saying, complex at a simple level because it's repetitions of a simple rule. There's an interesting um, concept or idea that never caught on for doing image compression using those types of things, where you would try to find for a given image, you would try to find a simple function that if you iterate enough times would eventually give you that image that you're looking at. Turns out it's not always easy to find that simple function that you could iterate to get an arbitrary image or approximate an arbitrary image. But it's an interesting, yeah, it's an interesting field. It's an interesting argument about whether those are genuinely developing complex information or whether, I mean, it's clearly just a simple rule. So it's simple information that generated those, what appear to us to be complex scenes.
Well, I like them because they're sort of each of the objects, each of the agents sort of knows about its immediate surroundings and it knows about its neighbors, for example. And if its neighbors do something, it reacts. And by that kind of reaction and being aware of your neighborhood, the object either lives or dies or does something. It behaves somehow. And you can build machine learning methodologies to follow rules like that. And it can, you can set up the scenario and it can learn the rules to the game, which is kind of interesting. And in a sense, these kinds of things are a way to set up the simulation to try and learn the rules of the game. if you will, I think.
Yep. Yeah. Yeah.
But
And
there's a lot of interesting things. I think we tend to be analysts. We tend to like observe. We like take a sample of something and we measure something and we say, oh, look, it's different in this group than in that group. But we actually very hesitant to sometimes do simulations like this and try and actually put things into motion, which is really kind of fun. I think can be exciting and give one some insight into the actual underlying complexity, whether it's simple or it's some really horrible, partial differential equation, for example. But for the most part, I kind of wonder whether or not there aren't simple rules that give rise to some of the behaviors and the things that we see dynamically that are, say, part of, I don't know, part of a genome, part of a transcriptome that we haven't fully appreciated yet.
yeah. Yeah. And I used to be um, not very big on the agent-based models because you can't solve analytically for what's going on. Kind of like you're saying, there's this randomness part of it, there's the complexity. But the approximate Bayesian computation, one of the really amazing things about it is you can use it to infer general rules about the inputs and what they lead to, even though the intermediate behavior can be extraordinarily complex.
Well, this is really fantastic. Well, Bill, I think this has been really a great conversation. You've really given me a lot to think about, in just how one goes about thinking about these models, about some of the things that one needs to consider. Obviously, NetLogo looks like it is a real interesting resource. I wasn't familiar with it, and maybe some of our listeners aren't as well. And so I'm hoping that people will go there and take a look at these things and try and play around with some of these models because they look like they might be a lot of fun. And thank you so much for sharing your experience with us and your wisdom on kind of these methodologies. It's very exciting.
Thank you, Jack. Thank you for the time to interact and talk and share, and hopefully things are useful to people. And I'm glad to ask any follow-up questions if people want to look me up and ask about things.
Wonderful. Well, thank you so much, Bill. Enjoy your weekend, and we'll look forward to seeing you around here around UVA campus. For everybody else, have a wonderful Friday and a wonderful weekend, and we will look forward to seeing you next time here at the Foundations of Biomedical Data Science Seminar Series. So thank you.